Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another awesome episode of our show. Bring you a really great guest today involved in creating a better tomorrow for all of us. Some really interesting science and tech we're going to be getting into today. Today, we have the honor of being joined by Dr. Rasha Hammamieh, Director of the Medical Readiness Systems Biology Group at Walter Reed Army Institute of Research, where her lab seeks to apply integrative biology approaches to various diseases of military relevance, such as post-traumatic stress disorder, traumatic brain injury, pain, and biologic responses to so-called polytrauma and austere environments, ranging from everything from hypoxia, radiation, microgravity. And the majority of Dr. Hammamieh's work is to provide an understanding and ultimately to develop countermeasures for various diseases and injuries in support of our service members. Currently, the lab is assessing the effects of spaceflight conditions on board the International Space Station on both health and disease. And Dr. Hammamieh's team has multiple biologic investigations sponsored by the ISS National Laboratory, investigating the effects of microgravity on things like the healing of both bone and skin wounds. Dr. Hammamieh has authored 150 publications, 3 book chapters, a co-inventor on different patents related to the biomarkers of immune dysfunction, related to chronic stress. She did her early undergraduate work in the area of pharmacy at Damascus University in Syria, moved on to do a Ph.D. in biochemistry from Georgetown, and wanted to have with us a lot of really interesting themes to get into today. Dr. Rasha Hammamieh, thank you so much for taking the time to come on the show.
Thank you so much for having me. This is very exciting. Very happy to talk.
Yeah, I'm really excited about all these themes. Would love to, as we typically do, start off by handing you the floor for a couple minutes just to talk a little bit more about your background. I'm always intrigued, as I'm a pharmacist myself, of how someone made the leap from pharmacy into biomedical research and then obviously into what you're doing now. But love to hear, a couple of minutes of your background story as well.
Sure. Thank you. So I was grew up in Damascus, Syria, as you mentioned. I got my degree in pharmacy from Damascus University, and then went ahead to do a master's degree in clinical biochemistry. And while I was doing this, I'm like, you know what? I don't think and this is what I like about doing these kind of extra not work but extracurricular activities to really understand what exactly you'd like to do. So when I started doing my master's degree, I thought that, I don't think I'm a fit for being in a pharmacy or, prescribing, doing drugs, dispensing drugs or medications. So one of my mentor, who's a friend as well, who had a PhD from Georgetown University, was like, you're doing very good here. Why don't you just go ahead and apply for a PhD program? So that was it. Actually, I submitted my application to Georgetown University, and I was like, I'm not gonna get it. Might as well just do it. Submitted my application, and months after, I got the letter saying that I was admitted to Georgetown University. That was the only school that I submitted my application for, and it's very surprisingly very happy, very fortunate to be able to get into a PhD program at Georgetown University. So. I went to Georgetown. That was like almost 30 years ago. Wow. 30 years ago, worked in the lab of Dr. David Yang, chemistry department. And it was very basic research that we were doing, basically understanding the function structure of what we call the amino acid tRNA synthetases, understand how they bind to the different motifs from both proteins and DNA and tRNA, and understanding the mechanisms of the binding. And that was actually the first time that I got into what's called the modeling and simulation. It was fascinating actually to see how the molecules move in a computer. And that's what got me more interested in how we can apply this computational biology to biology, or computer science to biology. So I got my PhD from Georgetown, and while I was finishing my PhD, actually one of my colleague and friend who was just finished his PhD and started working at Walter Reed Arbus in the research, said, you know what, there's an opening at Walter Reed, has nothing to do with tRNA or with what you're doing, basic research, but maybe focused on biomarkers. If you'd like to join, I was like, yeah, definitely. So I applied to the job, a postdoc position at Walter Reed Arbus in research with Dr. Marty Jett. And I got the job there as a postdoc, very different from what I was doing at my PhD, which is trying to look at some of the biomarkers that would relate to breast and prostate cancer. So, and that's what got me into Walter Reed, and since 1998, I have been at Walter Reed Arbus in the research. So I think that's the beginning of my career there at Walter Reed.
Yeah, absolutely.
So, one of the things I would like to mention is at Walter Reed, it's known to be the premier medical research facility for the DoD, where they do not only they do under everything that's of military relevance, not only psychological health, TBI, blast exposures, neurotrauma, as well as behavioral biology, military psychiatry, but also there is a huge emphasis on infectious diseases, emerging infectious diseases, diarrhea, wound infection, other infectious diseases, for not only for diagnosis but also for intervention. And this is why I love what I'm doing here at Walter Reed, and I've been here for 30 years.
It's really interesting because my understanding, and in the past, we profiled Colonel Victor Suarez, and he, sort of gave us sort of the introduction to the Army's Medical Research and Development Command. And my understanding is Walter Reed Army Institute of Research is under that umbrella. And again, as you were just mentioning, the scope of work that you've been involved in there, and then and we'll touch on a couple of those, but then we'll get into medical readiness systems biology, what that's all about. But I'd love to talk about a couple of the things that you sort of worked on earlier in the day. You mentioned infectious diseases, and I think some of, and again, the theme of sort of systems thinking comes into all of your work. And looking back, say, this was about 20 years ago, you're involved in a really interesting, it was called, I think, Project Normal. It was kind of somewhat similar to the ECHO project at DARPA we profiled, where basically you're looking at the complex sort of genetic gene expression dynamics that change when we are infected by things. We normally think, oh, I get infected and I get sick. But no, there's a lot of these interesting changes that happen quite early on that make potential diagnosis something that could happen a lot earlier. Talk a little bit about, because we don't always think about the sort of the systems biology component of infectious diseases, but this was an area that you spent a lot of time on early on. And I'd love you to talk a little bit about that. I think it's important.
So yeah, I was always fascinated actually with our system, with our body. When you think about it, we are the product of our genomic makeup and the environment we're exposed to, right? And our system is a marvel of these complex interactions. Cells are interacting, molecules are interacting, proteins, metabolites, even genes, and you add to it now the different organs are talking, and then on top of all that you add the gut microbiome and the different microbiota that's in our different areas in our system. So the fact that all of these complex interactions, we really need to find a way to capture all of this happening at one point in time. And this is why we're looking at applying this, what we call integrative omics, screen every single molecule in the system in response to a specific stressor, whether it's an exposure to a pathogen, a radiation, exposure to chemical toxicant, or even exposure to a traumatic event. We're the sensors of what we're exposed to, right? So, but then you can add to it your genomic makeup. So our genomic makeup will make us, what makes us more or less predisposed. And this is all about probability, right? So it will make us more or less predisposed to a specific stressor. So you and I will be exposed to the same either traumatic event or same exposure, but you and I will develop different outcome. And this is why our part here at Walter Reed, the medical readiness systems biology, is understand not only what happens when we get exposed to something by screening all these genes, proteins, metabolites, even the gut microbiome, to understand mechanism, identify these pathways and networks that are highly affected by this specific exposure. This will help us not only in developing, later on developing diagnostics panel for specific stressors, but also understand mechanisms. And this understanding this mechanism will help us develop novel interventions by knowing what are the networks and the pathways and the genes and proteins are highly affected by that specific exposure. And this is why we established what we call the medical readiness systems biology, not only to apply this integrative omics, but eventually get into what we call precision medicine, precision psychiatry. And again, there is not one disease that fits all, especially with the psychiatric help. PTSD, for example, and maybe we can get to this later, is not a simple issue. It's a very complicated issue with a lot of comorbidities. And again, you may develop sleep disturbance due to PTSD, and may develop metabolic disorder. And this is why we're trying to apply this integrative omics to see exactly what happens when you develop PTSD, for example, or you get exposed to a chemical toxicant or radiation, for example.
Off of what you were just saying, and we'll get to PTSD and some of the other indications that you're studying in a minute. But when it comes to this theme of medical readiness systems biology, so I was reading through a little bit on the website that it's sort of, as you're saying, it's disease agnostic. You're looking at all the sort of the multi-omics tools, and ultimately, I was sort of taken by sort of some of the definitions, in the sense that parallel a little bit of what we were talking about with the folks at DITRA a couple of months ago, that unlike where in the past, say for infectious diseases, we may have looked at a countermeasure for Ebola or a countermeasure against a specific chemical agent. In this particular case, it seems like you're going quite broad, not just looking at what potential stimulating agent is, but what are the underlying components. And I know you've done a lot of work in the area of the immune system and inflammation. What are some of sort of the core areas that you look at, and when you take this disease agnostic approach in sort of understanding the sort of the core of the system itself, and then of course we'll get into specific programs in a little bit.
Sure. So just to let you know, the mission of Walter Reed RMC Research is to develop products for the service member. So, and the product can be knowledge product, or can be a real product, not a real product, something that a tool or something. In our case, what we're trying to develop is this knowledge product that will help the warfighter, will help the service member, and not only the warfighter and the service member, but also the public. So understanding, for example, in the case of, let's say PTSD, you're exposed to a traumatic event, and we do not know, we're not sure whether the psychiatrist is like, you're borderline, we're not sure whether you develop PTSD or not. In the case of PTSD, the gold standard is the scoring system, right? Where there is a questionnaire, depending on how you answer these questions, you will be diagnosed with PTSD or not PTSD. And what we're trying to do is, in the case of PTSD, for example, look at blood biomarkers, which is much more objective than the way of the scoring system. Can we use this knowledge product that would be an aid in the diagnosis for PTSD? And again, this helps not only for the service member, but also for the public. The core aim or the goal of our not only Walter Reed, but mainly our program, our branch, is to develop these knowledge products. To use we do what we call, we call it fishing expedition, right? We don't know what we'll find. We know that interleukins were and there's going to be inflammation in PTSD. We know that interleukin cytokines will be going up and down all the time. But can we find something novel that we don't see usually with like what we not the usual markers, right? Something novel, something new, and this is why we try to apply these tools, and you add to it, now with AI, ML, and deep learning, the sky is the limit. Actually, there is no limit anymore with applying this AI, ML, the deep learning to the biology and the big data to not only help developing these diagnostic biomarkers, but also help developing novel therapeutic intervention.
Mm-hmm. Go deeper into that, because as you were mentioning, when you look at something like, and I'll put this sort of into the omics of PTSD, and again, looking at some of your publications here, and we'll put links to all this in the bio of the show, but you've written about everything from mitochondrial DNA circulating levels in the blood, glucocorticoid sensitivity. You did this major review just a couple months ago on epigenetic dynamics of PTSD, and then paper on actually using machine learning to sift through this amazing mountain of data. Talk about a little more, because I think it's important to understand, as you were saying, you're not looking for one thing, you're looking at lots of things, the known and the unknown. So talk us through some examples there of that particular work. I think it's a nice proxy for the rest of what's going on.
So one of the things that I don't want to take credit for everything that you mentioned. One of the things that I'm so blessed with is I'm surrounded with some of the brightest and most hardworking scientists, not only in the DoD, but not only in the DoD, but in the US. We have 8 computational biologists in our group who are doing data analysis, data mining, data integration, and just deep learning. And this is actually what helps a lot. Biology used to be, do a PCR, look for a gene, look for a protein. Not anymore. Biology is math, computation, physics, you name it, all of the above. So some of these publications that we had, and I'll get to the methylation agent, for example, later on, but is some of our computational biologists are literally applying this high throughput data mining, data analysis, and deep learning to really, and again, in complex issues such as PTSD, there is a lot going on. There is a lot of information that would be gathered not only from gene expression, but also metabolites, proteins, protein expression, even the gut microbiome. And again, trying to apply these big data approaches to biology to help, again, not only identify tools and panels, but also mitigate the outcome. And the publication that you mentioned, mainly applying AI, and now we have a huge project actually on deep learning, where we're looking at what most of the publicly available databases on gene expression profiles and methylation profiles for PTSD, and get at all of these databases, try to mine all of these databases, not only with the data that we have, but also whatever is available to again develop more trust, I would say, reliable biomarkers of PTSD.
And moving from a condition like a PTSD, where no doubt you're able to capture a tremendous amount of info, to something like your work in microgravity, obviously we have the International Space Station lab. We have Polaris Dawn just came back to Earth a couple of days ago. Maybe not as much data, but again, you've been publishing, and your team, quite extensively on everything from immunodeficiency dynamics during space flight, endothelial cell, omics, fracturing of bone and bone healing. Talk a little bit about, this is just an exciting topic on its own in terms of understanding the biology of microgravity, but at the same time, when you don't have as much experience yet, I mean, hopefully we'll have a lot more coming back from low Earth orbit and stuff like that in the coming decades. But talk a little bit about that, because I think that's an interesting sort of contrast to something of PTSD where we have much more data available.
Exactly. So this our effort with NASA and the International Space Station started more than 10 years ago. It was actually a tasker that came down to us in an email saying, if you have any idea on space biology, please reach out to us and submit a proposal. And that was so there's something that's called the DoD Space Experiment Review Board that reviews all proposals, not only biology, but engineering and satellites and others. So at that time, one of my colleagues, Mr. Navarro Chakraborty, was like, let's give it a try. And one thing I didn't tell you is I always wanted to be an astronaut. It didn't happen, but but I was like, what? Let's give it a try. So we submitted a proposal to assess the effects of microgravity on infectious, because it's been actually known before we sent our experiment that spaceflight environment not only affect your immune system, but also it increases pathogen virulence. So we submitted a proposal to the DoD Space Experiment Review Board, and I think at that time it was the only biology among maybe close to 50 or 60 experiments. We submitted that proposal, and we were invited to come and defend it. And long and behold, we were ranked, and once you ranked at the DoD Space Experiment Review Board, that means that you get a slot, you can go ahead and manifest your experiment. And that means the involvement of what's called the DoD's Space S program, that were the liaison between us and NASA and the International Space Station. This is how the journey started back in 2010, and we sent our bioreactors actually on board Atlantis at that time, the last shuttle actually, the last NASA shuttle. I can tell you it was a once in a lifetime experience. And I'm still, every time I talk about our experiments into space, I get very excited. So since then, we got this relationship with NASA and the DoD's Space Experiment Review Board and the DoD's Space Test Program. And started, coming up with new ideas. The latest were the effect of that spaceflight environment, microgravity, radiation, and stress together on bone healing and on wound healing. So our experiment on the bone healing involved sending mice into space with bone fractures. So this was in collaboration with Dr. Melissa and Cassine at Indiana University, the orthopedics department. They're the experts. We're not experts on bone healing. And this is what we do in our lab. Actually, we reach out to the experts in this field. We do not reinvent the wheel. So we talked to Melissa at one of the military health system research symposium. And let's just go ahead and submit this proposal and see what happens. So we submitted the proposal, again, got ranked, and we were able to manifest that first experiment. And this was the first experiment with bone fractures that were sent into space. So we sent 40 mice to the International Space Station. We had the mice in two different groups. So we have one group, a control group with bone fractures. We had one group with bone fractures with no fractures at all. And then we had two groups with bone fractures with two interventions. One of them was BMP2, and the other one was TPO, which was developed, which was used by Indiana University for interventions for bone healing. So, and that when we sent the mice into space, astronauts actually were trained to do some animal interventions. So they would take the mouse and they will do the cardiac puncture, get the blood, and then get the bone for us for assessment, and then send us the carcasses back where we did some whole genome analysis, because these are very precious animals. coming back from that spaceflight environment. So what we wanted to do is not only assess bone healing and space environment, but also assess how that space environment would affect the brain, the gut microbiome, the lungs, you name it, and the eyes, everything in the system. So from these mice, we collect thousands of samples to really assess the effect of spaceflight environment on our system. So we finished the bone healing one, and then we recently manifested the wound healing study. And this was in collaboration with the Burn Center, Dr. Jeff Schaap at Washington Hospital Center. And again, they're expert on wound healing. So the astronauts actually were trained to do wound incision on orbit. And this is the first animal study in space where there is surgery, there is wound incision on orbit. So we had to do a demo experiment before we manifested this specific experiment. And again, we sent 40 mice into space, and the astronauts did the wound incision on these mice. And then we collected these mice back, and we're doing the analysis as we speak. And we presented actually a couple of meetings and some of our findings, both in wound healing and wound healing.
Really?
So that's yeah, it's been very exciting.
Yeah, it's again, I know it's a new area for in the sense that, there's not as much data around as other things, but what we're going to be getting, I think it's rather exciting that thinking out as we get more back into space than we've been, to be able to collect this data and then do the types of multi-omics work that you're talking about. Again, thinking of the space station and thinking of things like NASA's twin study on the sort of the aging and then methylation patterns and all that stuff that was in the press a little while ago. It got me looking at a paper that you did. This was 2021 in Nature Molecular Psychiatry. You're a nice for the team here. Lee Hood, who we had on the show a couple months ago, is involved with this one, entitled a DNA methylation clock associated with age-related illness and mortality is accelerated in men with combat PTSD. And here, this is interesting because you're bringing in several of the things we talked about, but what you do here is you take the premier sort of epigenetic clock tool, this DNA Grim age, and you modify it a bit to create something called Age Excel Grim, looking at, again, the connection to how one can speed up the Grim Age clock in trauma-exposed veterans. And I'd love for you to say a little bit about this as well, because, and I want to ask a question about clocks in general, but talk a little bit about this part, because I think this brings several of the themes together in a really interesting way, and a little bit of what you've learned.
So this is a study that was led actually by Dr. Routine Young, one of our lead computational biologists here at Walter Reed, and he literally went and met with Dr. Hogarth, who was, a lead on the clock and methylation aging. He literally traveled there to meet with him and talk to him about some of our studies, and he came back and started doing the analysis of, let's understand. Try to see whether there is an accelerated aging in those service members that come back and have PTSD, right? And long and behold, he was able to show that yes, there are some accelerated aging. DNA methylation clock is or the Grim age, is fascinating. It's a good measure of your environment. For example, stress will induce accelerated aging. Junk food, for example, will induce accelerated aging. And what's nice about it, it can be reversed with good lifestyles. Exercise, a healthy diet can reverse this Grim age and accelerated aging. And it's something that we thought that would be fascinating actually to show that yes, even though there is some accelerated aging, but it can be reversible. So if we check your epigenetic age and we see, yes, there is some accelerated aging, what are the tools that you can use now to reverse that outcome. And maybe, the telomere length and telomerase activity used to be the gold standard for this, but now we're adding something to it, which is this epigenomic or epigenetic aging. And again, that can help you not only to see, yes, there is accelerated aging due to stress or due to PTSD, but also it can be reversed with, diet, with good diet, with exercise, for example, with healthy lifestyle. And we're still doing a lot of work actually on the specific aging, not only in PTSD, but also in what we call acute stress disorder, which is, very quickly after exposure to a traumatic event, we start looking at your methylation or epigenomic aging and see whether very quickly we can identify, yes, there will be some accelerated aging due to exposure to this traumatic event. I would love to do other studies on aging and in response to radiation, for example, or in response to exposure to austere environment, like heat stress, cold stress, even hypoxia. But we go with what funding we're able to usually get. So, but we have the capability when we have the tools and the samples to do it. So we would love to do it if possible.
Mm-hmm. Side note related to that, because I know that you have some patents of your own where you look at things like immune dysfunction, immunosuppression, and again look at sort of the combination of in this one patent I looked at sort of various chemokines and interleukins and things like that. Is there sort of clocks? I don't know, call them clocks. What are the best term for it? Because I know there's sort of debate, is it epigenetic clocks? Is it proteomics clocks? Is it, in this particular case, these really interesting sort of immunometabolites? What is the ideal sort of clock, in your opinion, or set of clocks in terms of sort of this systems approach? Because there's so many out there that are sort of, different approaches, and I'd love to just get your perspective, because I know you've done your own work in patenting in this particular space, which is pretty cool. Just like as sort of a side topic, I'd love to sort of get your perspective on that.
Yeah, so I'll start. I want to talk about this stress study that we did way back. We got the patent approved for, but before I do that, the answer is there is no magic answer actually to this, and we're still scratching the tip of the iceberg on this, because especially same with the microbiome. There's still a lot of unknowns about this, and we do need huge numbers of samples to get to the answer that we're looking for. So going back to the stress study, this was a study that was led by Dr. Marijet and Dr. Saeed Murhi, and this started more than 13 years ago actually, where we collected samples from rangers before they started ranger school, and then during the ranger school, and then at the end of the ranger school. And what we found was that, so at the beginning and the end of their training, ranger training, again, collected samples before and after, blood samples, and not only we assessed, we did the whole genome analysis. At that time, we used to use microarrays, not sequencing. But so we did the microarrays on before and after, and we did show, and Saeed had shown actually that there is a decrease and there is an immune dysfunction, bottom line, and those individuals after, and that's mainly due to psychological, physiological stress, all the stressors that these rangers undergo. One other thing that we did in Saeed actually was expose their blood samples to pathogens before and after the training. And again, we did show that there are differences in responses to these pathogens. So inflammation, immune response, immune dysfunction, a lot is happening due to this physiological and psychological stress. And we can show it also in individuals that have PTSD, for example, that inflammation is an issue, immune response is an issue also, and so there is a lot, this inflammation and the immune system plays a huge role in psych health, bottom line. So there's a huge connection. We don't know whether it's the gut microbiome, this gut-brain axis that plays a huge role, but inflammation again is one of the markers of site health.
As we're talking, and I just want to make sure the audience understood this, because you're not just focusing on PTSD, but there's this related condition that we maybe don't hear as much about, acute stress disorder, which my basic understanding, it happens a lot earlier than sort of the PTSD dynamics. And I wanted to highlight that because your most recent paper, which was exploring metabolomic dynamics in acute stress disorder, amino acids, lipids, and carbohydrates. It got me thinking, and I'm going to loop back now to sort of the early part of your career, because you started out, interestingly enough, doing research on cancer and the sort of the lipid biology and the arachidonic acid cascade and all that bit. And I sort of like to come full circle here. I'd love you to talk about the current work, but again, thinking back to sort of, I guess, one of the more ultimate systems issues, namely cancer, where we've maybe gone highly in a direction of very targeted therapies, but again, thinking of the fact that there's still major systems behind cancer nowadays that we maybe haven't explored as much. Would love for you to sort of loop those two themes back together, because it's where you started and it's still what you're doing now, and love to make that connection.
Yeah, so it all comes back to the complexity of our system, right? And again, the usual way, the conventional ways that was way back of doing one thing at a time, one gene, one protein at a time, is beyond us. So and understanding, appreciating how complex our systems are. And again, this different crosstalks between the cells, the organs, and the gut microbiome and the gut-brain axis. All of these plays a huge role in our responses to stressors, whether it's cancer or PTSD or whatever. So, and this is one of the reasons why in every project we have, we try to collect. So we know, for example, the PTSD, the brain is the main outcome. But we publish showing that the heart is affected. There's a soldier heart. In exposure to radiation, we did show that the gut microbiome is affected. It's not like one organ that's been affected in exposure to plague, for example, to asbestos. We've seen changes in different organs, not only the lungs, or the blood. So just appreciating that the complexity of our system, and that there is a lot of interactions that are happening, and there is some kind of a symbiosis, a balance within the system, and an exposure to a pathogen or radiation or cancer or even a traumatic event will affect this balance. And that's when, everything starts talking, everything starts working together to mitigate the outcome. And again, whether it's cancer or PTSD or exposure to radiation, the idea of one organ has been affected is not what we're seeing right now. Is that there's a lot of these interactions between the organs, between the and again, I keep going back to the gut microbiome, because I think we're still scratching the surface on the gut microbiome, and there is a lot that can be used for the microbiome, not only as sensors for exposure, but also can we manipulate the gut microbiome to mitigate the outcome. So yeah, it's again in science, every day I've been working for 30 years, and believe me, every day I learn something new. It's just amazing.
At least for the microbiome and virome related episodes we've done, it's clearly an extremely uncharted territory, and now realizing sort of the extent to how much of these organisms live within us symbiotically, and there's so much there still to learn. And I think with the type of stuff you're doing, plus the machine learning tools to be able to crunch it all, it's a very fertile ground for future discovery. ask you about one other thing while we have you today. I just a couple of weeks ago, I did an episode with Dr. Francis Collins, and just retired as head of NIH, and they were talking about his career, and one of the things that he thought is a space that is extremely fertile that we haven't put a lot of effort into yet is sort of the genomics, the pharmacogenomics of addiction and overdose and sort of the whole opioid epidemic thing. I know you published about this as well, and this is when we talk about stressors, this is a major stressor, whether we're talking about the biology of addiction or unfortunately the connection to suicide. If you could just say a little bit about what you're up to on that front at MRSB, I think that would be really important for our audience.
Yeah, so we're doing actually a study with Dr. Jonathan Boyd at VCU, and this is a fascinating study. And so they're exposing animals to fentanyl and other chemical toxic, and the idea is, so what they're trying to do is they're using some imaging tools and they're injecting their mice with markers that would glow in inflamed organs. So the idea is, can we look at inflammation in the different organs, in fentanyl, for example, in exposure to fentanyl, and then take these organs and let's do this multi-omics to understand exactly mechanisms of what happens in the different organs in exposure to fentanyl. So Dr. Boyd at VCU has been exposing these mice not only to fentanyl, but to other chemical agents. And again, we're finding changes in different organs, which is, fascinating. And so it's not only the brain, but also you can see it in other organs and tissue in our system. And again, going back to pharmacogenomics, and which is maybe not the topic for now. Pharmacogenomics is also should also understand the complexity of our system. We're very different, right? And this is why SSRI, for example, serotonin receptor inhibitors, they're not very effective in all patients, right? And this is mainly because due to your genomic makeup. So again, the responses to fentanyl, will be different because of your genomic makeup. So intervention and pharmacogenomics, I think we need to really rely on and appreciate the precision intervention, right? Precision psychiatry and precision medicine overall, because again, this genomic makeup will play a role in how you respond to a stressor and how you respond to a specific intervention. And in the case of addiction, unfortunately, I'm not an expert on addiction, but I can tell you that with what we're finding with exposure to fentanyl, for example, is that it's the whole system that's been affected. It's the gut microbiome is affected, the brain, the heart, the liver, everything is affected by exposure to fentanyl. So again, I keep going back to to really appreciating how complex our systems are, and we really need to capture by applying this holistic approaches or integrative biology approaches to different stressors to understand really what happens in the case of a specific exposure.
Really fascinating portfolio. So much opportunity to go further and delve into all these areas at a level that we just haven't had available to us. And it's exciting to see sort of this as mentioned, this sort of this disease agnostic approach to really finding out what else is out there in terms of these mechanisms. Really, really great stuff. So what else is coming up for you? I know that you are out on the conference circuit quite a bit. I think you were at the American Psychiatric Association recently. I think there's something coming up at Carnegie Mellon in terms of the microbiome that you're going to be speaking on. What else is happening for you in 2024 that we should know about in terms of conferences, talks?
So a lot actually is happening next year in the next 18 months, I would say. So we're involved with DARPA on 3 different programs. Our group is being used, and RARE, actually, Walter Reed Armstead Research is being used as what's called an IVNV, which is independent validation and verification for the DARPA. So we have 3 programs that we just got the approval to be involved in as an IVNV. One is Somnus, one is Shield, and then the Cornerstone. Three different groups. This will keep us busy. This was I was just saying it's a good problem to have. It will keep us very busy. But we're also submitting actually now to the FDA, and this is something that I forgot to mention, which is so we were able to identify within the first 24 hours predictors of the development of sepsis days before onset. So this is in burn patient. This is with Dr. Jeff Shop at the Washington Hospital Center. And the idea is in burn patients, can we, within the first 24 hours of admission to the ER, can we predict who will develop sepsis and who will not? And this is very big. I think it is big because the usual methods will predict death in sepsis. What we're trying to do is who develops sepsis, and this will help with the intervention. So going back to what's waiting for us next year is we just did a pre-submission to the FDA, and we submitted a patent application on the panel for sepsis, and we have one company that's interested in licensing our tool. We also submitted, working on the submission for the FDA for the PTSD panel, diagnostic panel, showing that PTSD is not a one disease fits all. There are different biotypes of PTSD, and this is what we're trying to get into precision psychiatry. So there are different, just looking at the data, you can identify different biotypes of PTSD, and this is what we're submitting also to the FDA. Other projects that we're involved in right now are, how we can, how the host is responding to austere environment. Can we predict who will develop acute mountain sickness, for example, at sea level? Can we look at your gene expression? This is in collaboration with the U.S. Army Institute for Environmental Medicine Research in Syria, Dr. Beth Barrowman, to show how at sea level, how can we look at your mRNA and microRNA and genes? Can we predict whether you develop acute mountain sickness or not when you go to high altitude? Other austere environment we're working on are heat stress and cold stress, also in collaboration with this area. So there is a lot, and I think this next year will be very, very productive for us. We have a few submission for patent application for blast exposure, biomarkers of exposure to blast, as well as live TBI. But again, it will be a very busy year coming.
You won't be bored. This is a really amazing portfolio, and I just, I enjoy listening to you talk about it, and I enjoyed reading all about you and your papers beforehand. But I, again, wishing you the best, you and your team, the best with all that you're going to be doing in the coming year, and in further understanding these unique conditions that ultimately, benefit our service members, but also all of us. And the knowledge that you generate. So really great stuff, Rasha. I enjoyed the discussion. Again, for everybody that's going to be listening to this episode of our show across the various podcast networks, or who will be watching on the YouTube channel, again, you've been spending time with Dr. Rasha Hammamieh, Director, Medical Readiness Systems Biology, Walter Reed Army Institute of Research, doing really amazing things to understand and ultimately develop countermeasures for these diseases and injuries in support of our service members. Rasha, I want to thank you for taking the time out of your schedule. I know we're coming up to a hard stop, but want to thank you again for your time. Thank you for everything you're doing there at the organization. And as we like to say on our show, thanks for helping to create a better and safer tomorrow for all of us via the type of work you're doing. Really a great story, and I look forward to continue to follow you and your team.
Thank you so much. Appreciate it. Thanks a lot for giving me the chance to talk.
Really great having you.
Thank you.